Welcome to the third video on genetics. So far we have looked at introduction, we looked at variation and chromosomes, then in another video we looked at cell division. In this video we are going to look at a very interesting part of genetics called monohybrid crosses. We are going to become detectives after doing this. After that we are going to take a look at blood groups, sex linkage and mutation in the next video. With that being said, let's get started. First, we're going to acquaint ourselves with the language of genetics. Some terms I introduced them in the first video, but we're going to have a fresh perspective. The first is DNA, which is short for deoxyribonucleic acid. So, what is DNA? Deoxyribonucleic acid. This is the hereditary material in organisms. So remember, genetics studies inheritance, how we inherit certain features from our parents, or how we transmit certain features to our young ones. So the material that carries this information that is transferred from one generation to another is what is called DNA, or deoxyribonucleic acid. With that being said, the next term is a gene. So a gene is a portion or segment of DNA controlling one particular characteristic. So DNA are all different features that we have to transfer, or that are transferred to the next generation, but a particular portion or segment or section of DNA that controls one particular characteristic. For example, your height, the one that's going to transfer your height to your young ones, is a gene. Okay, so a gene is just a portion that is concerned with one particular characteristic. Again, I'll be using the word trait to mean characteristic. I'm used to trait rather than characteristic, so sometimes I'll be using the word trait to mean characteristic. So when I say trait, characteristic, I mean things like your height, your voice, your skin color, the color of your eyes, and your mungulu to be included. With that being said, the next word is allele. So an allele is an alternative form of a given gene. So an allele is just an alternative form of a given gene. So remember, a gene can be something that determines your height only. Okay, so there's a gene for height. So if there's a gene for height, there might be different heights. So there is a gene for tallness, and there is also an alternative form of that gene. In short, an opposite form of that gene. So if there is a gene for height, then there is another gene for shortness. So if you have a gene, its alternative form, in short, two genes, each is an allele, because they are alternative, they are competing. One says this, the other says this. One says this, this person should be tall. The other one can be saying this person should be short. So that's how genes are. They usually have alternative forms or opposite forms. So for example, height, we can have tallness, shortness. Skin color, we can have dark, light. Eye color, we can have blue, brown, is it coffee, black, and the likes. So an alternative form of a given gene is called an allele. So each sexually reproducing organism has two alleles for each of its characteristics. Like I said, tallness, shortness. So this should stick. So each sexually reproducing organism has two alleles, which means two opposite genes, or two contrasting genes, or two alternative genes. But there are situations where we have multiple alleles, such as blood groups. So for blood groups, we have different blood groups, which each of the blood group, the type of blood group, has got genes that determine it. So the different combination of genes will produce a given blood group. So there is a gene for blood group A. There is an allele for A, an allele for B, an allele for O. So the way you combine these three alleles determines the blood group of a person. So multiple alleles is a situation where we have more than two alleles. Okay, so normally in sexually reproducing organism we have two alleles. For example, tallness, shortness. But multiple alleles is where we have more than two. Next we have chromosome. So a chromosome is a structure wrapped in a protein and on which genes are located. So these genes which we are talking about, which are a segment of DNA, are usually located on a chromosome. So a chromosome is going to have sections that have genes. Okay, so a chromosome, what you should take is a chromosome is where genes are. Genes are the ones that determines the characteristic. Then alleles are different forms of a gene. Multiple alleles is a situation where we have more than one allele, and the DNA is the material for inheritance. So chromosomes carry inherited information from one generation to the next. Okay, so chromosomes carry inherited information from one generation to the next, which simply means chromosomes carry genes which of course is representative of the DNA. So chromosomes carry DNA because DNA is the hereditary material. So chromosomes are the one that carry this DNA. So we're talking about genes being segments of DNA and these genes being on chromosomes. Simple as that. So in our studies we'll be talking more of chromosomes and alleles or genes. DNA not that much. Next, we look at a term called genotype. So genotype is the genetic characteristics of an organism. So an organism has two types of characteristics. The first is genotype. Genotype is how genes are combined to produce a certain characteristic. So for example, your height. So your height depends on how the gene for tallness and the gene for shortness were combined to give that height. So sometimes there are situations where a person is tall but inside them they are short. So picture this scenario where we have a tall man and a tall woman. Then they have a short child. So how is it that there's a short child? It means both of them had some shortness in them but hidden. So you cannot tell by looking or by observing. You can only tell if you do a DNA test, so to put it. But if you know their genotype, what makes the kind of genes that makes up their characteristic. So genotype is just the genetic characteristics of an organism. So genotypes are written using a pair of letters, each representing one allele. So for example, if let's say we're talking about height, or I don't know what A can represent, maybe let's say okay, A represents height, then capital A we can say is tallness, then small a we can say is shortness. So this is an allele and this is an allele. Another situation is this is an allele and this is an allele. So if we have these two alleles, let's say capital letter A is for tallness, then we mean that this organism is purely tall. It has no shortness in it, as you are going to see. But this organism, we can say it has tallness and shortness. So it can be tall, but there is some shortness in it. So in our studies of genetics, we're going to be using letters to represent alleles. Then phenotype is the observable physical characteristics of an organism, e.g. height or eye color. So what you see, that's the phenotype, what you see with your eyes. So phenotype can be misleading. Okay, phenotype can be misleading. Someone can be tall but they are not purely tall because they can have a child who is short, as you are going to understand. Then another interesting term is dominant. A dominant allele is one that gets expressed in the phenotype to the exclusion of the other. So we go back to this part here. So here there are two types of alleles. One is called a dominant, the other one is called a recessive. So maybe we define a recessive before I can explain further. So for the dominant, it is always represented by capital letter, e.g. A, B, T, and so on. Then a recessive allele is one that is overshadowed in the phenotype by the presence of a dominant allele. So for a recessive, it does not appear in the phenotype. You cannot see it, you cannot hear it, and the likes. So it is always overshadowed in the phenotype because there is a dominant allele. But if there is no dominant allele, then you can observe it, you can see it. So in this case, in this situation of two capital letters, these are both dominant alleles. This is a dominant, this is a dominant. This other one, we have a dominant and a recessive. Okay, so this person might appear, if A is for tallness, might appear tall, and this one might appear tall, but this one is purely tall. There is no chance that this one can have a short child. But this other one, this one is tall, but there is some shortness. So there is a chance that this one can have a short child. So this allele that is that you cannot see when there's a dominant is called a recessive. Then this one is a dominant, which is always seen when present. So the recessive is always represented by small letter, like I said. The next term is homozygous. So homozygous is a type of genotype where both alleles are the same. So meaning both are dominant or both recessive. So when you come across the word homozygous, it means the alleles are the same. It can be both a capital S or both a small s. The other one is by the way, organisms which are homozygous are called homozygotes or pure breeds. Like I said, if someone, for example, if this was for tallness, then this one is purely tall. So organisms that are homozygous are always pure. So it can be purely short, purely tall. Then the other term is heterozygous, which is opposite of homozygous. So heterozygous is a type of genotype where the two alleles are different, such as one allele is dominant while the other is recessive. So a situation where we have different alleles, to the dominant and the recessive is called heterozygous. So e.g. AA, TT, and BB. So usually when they are stating homozygous, they will say homozygous dominant to mean that they are both dominant or both capital letters, or homozygous recessive to mean that there is a capital and a small letter. Then for heterozygous, they just say heterozygous. You have to know that there is a small and a capital letter. Organisms which are heterozygous are called heterozygotes or hybrids. Then there is a situation called codominance. So codominant alleles are two different alleles which are equally expressed in the phenotype when present. So in codominance, there is no dominant, there is no recessive. Both are expressed. You can see both. So if tallness had alleles which had a codominance phenomenon, then the allele for tallness and allele for shortness, when they are combined, they will appear to you'll be able to observe both of them. You'll be able to see that this person is tall and this person is short, which doesn't make sense because the allele for tallness is not a codominant allele. So a good example, there are some flowers which are called snapdragons. So they can either be red or white. Okay, so red is dominant, white is dominant. So when they are mixed, since they are both dominant, the allele is going to the offspring is going to appear to have both red and white. It will appear to be pink, which is like the midpoint of red and white. If I'm not mistaken, it has to be pink anyway. So just know that in codominance, both alleles are expressed. So another good example is blood group. Blood group A and blood group B are codominant. So if the mother's blood group A and the father's blood group B, the child is going to have blood group A, B, because both of them are codominant. Now we start the interesting part, monohybrid crosses. So monohybrid crosses are crosses used to determine the genotype and phenotype of offsprings. So if before you marry, for those who are not married, you have to know the genotype of your partner and your genotype for you to know the genotype of the children. You can tell how the children are going to be. So that's what we're going to start to be studying in monohybrid crosses, and this is where it gets interesting, like I said. So capital letter means dominant allele. In this case, determines tallness. Then small letter T means recessive allele. In this case, determines shortness, which is the opposite of tallness. So the word homozygous dominant, homozygous means they are the same. Dominant, which means both are capital. Homozygous, recessive, both are small because recessive is small, and homozygous means they are the same. Heterozygous means capital and small. Codominance, both capital. So with that terminology, let's try to attempt some questions. A homozygous dominant parent for height is crossed with a heterozygous parent for the same gene. Determine the genotype and phenotype of all the offspring. Okay, so first you have to get the genotype of the parents. So the first, let the gene of tallness be represented by capital by T, just by T, where capital T is dominant and small t is recessive. So for the parents, they have said there is a homozygous dominant, which means dominant capital letters, homozygous, there are two capital letters, they are the same. So we're talking about TT. Then heterozygous, capital T and small t. So let's see what kind of kids they can have. So what you do is you write the genotypes of the parents, TT, then we're crossing it with TT. So now we're going to be combining we're going to do different combinations of the alleles. This allele and this allele are going to combine like so. We'll be writing the genotype of the children here. So when this combines with this, we're going to have capital T's. Next, we're going to have this T combined with a small t there. We're going to have capital T and small t. Then this combining with this, we're going to have two capital T. Again, that one combined with a small one, we're going to have TT. So the genotype shows that we're going to have homozygous dominant, recessive, homozygous dominant, recessive. This is the genotype. If you're a doctor, you can tell. But what is it that people are going to see, which is the phenotype? They are going to see this one to be tall, this one also to be tall, because this one is recessive. You cannot see it even if it's there. This one is also tall, and this one is also tall. So what we're going to see, all the children are going to be tall, but two of them are pure tall, two of them are hybrid tall. Another example, heterozygous parent for height is crossed with heterozygous parent for the same gene. Determine the genotype and phenotype of all the offspring. So again, we let the gene for tallness be represented by T. So parents who have got heterozygous, which is TT, and another heterozygous, which is TT. Let's do cross C. So parents are these. So offsprings we're going to write here. So T and T, we're going to have TT. Capital T and small t, we're going to have T and small t. Small t and capital T, we're going to have capital T and small t. We always start with capital T. Then small t and small t, we're going to have TT. So this is the genotype. The phenotype, what is going to be seen is these are going to be tall. These also tall, because this cannot be seen when there is this. These are going to be tall. But this one, since they are all recessive, we are going to see that this is short. So 3 children will be tall, 1 will be short. So sometimes we can be told to express the genotypic ratio. That is the ratio of the genotype. So the ratio of the genotype is, we have 1 pure toe, then 2 which are hybrid toe, and 1 who is short. So the ratio of toe of pure toe to hybrid toe and to shortness is 1 to 2 to 1. Then for phenotype is 3 to 1. Toe to shortness, of course, is 3 to 1 because there are 3 toes and 1 short. Finally, there's this: heterozygous for white is crossed with a homozygous recessive parent for the same gene. Determine the genotype and phenotype of all the offsprings. So let the gene of tallness be represented by T again. Parents, of course, we have heterozygous, which is TT, and homozygous recessive. Homozygous recessive means they are the same. By the way, the homozygous recessive means that they are small letters. So we start crossing the offsprings. We're going to write here. So capital T, small t, it's going to give us TT. Capital T, small t, it's going to give us TT. Small t, it's going to give us small t's. Small t, it's going to give us small t's. So the genotypic ratio, we have 2 hybrid t's, a hybrid toe, and 2 short ones. So the ratios are 2 2, expressed in what is going to be 1 1. I'm going to show that. Then the phenotypic ratio is, we're going to have 2 who are tall and 2 who are short. So 2 children will be tall, 2 will be short. The ratio is 2 2, which you can express as 1 over 1, or it's 50% of the children are going to be tall and 50% are going to be short. So to get the percentage, you just get, for example, if it's for the percentage of children are going to be tall, 2 children out of the total 4 times 100, which is 50%. So we're going to look at something which is called the backcross. We're almost done. So a backcross is called a test cross. This is used when you, in this case, when an organism having a dominant phenotype is crossed with a homozygous recessive one in order to determine its genotype. In short, if you observe something to have a dominant phenotype, that is, for example, it is tall, but you want to know if it's purely tall or it's hybrid tall. So what you do is you cross it with a homozygous recessive. Then the outcome will tell you if it is dominant, if it is homozygous dominant, or it is heterozygous, if it is homozygous dominant or heterozygous. So what I'm trying to say is, someone is tall, then they have some children with the partner. So now you want to determine if these children belong to that person, and if that person has refused to do a DNA test, but you know that that person is, for example, tall. So what you can do is you have to do a backcross. So you get the gene for tallness. You're going to do 2 tests. In one, you're going to assume they are pure breeds. In the second, you're going to assume they are hybrid. Then you're going to do a cross with a homozygous recessive. So when you do that, we know the outcomes when you cross a homozygous recessive with a pure breed and a homozygous recessive with a hybrid. So let's assume they are pure breeds. So we are going to cross them with homozygous recessive TT. So here is what will happen. This time I'm not going to use a monohybrid cross. If you are fast, especially in paper 1, there's another cross you can do, which is called a grid. So on a grid, you just write for the parent one side, the other parent the other side. Then you start combining. So combining this and this, they are going to give us this. Combining this and this, it's going to give us this. Combining this and this, it's going to give us this. Combining this and this is going to give us this. So these are the expected children. So whenever a pure breed is crossed with a homozygous recessive, you're going to get all of them tall. So if all the children are tall, then you can tell that that person is a dominant phenotype. I mean, that person is a pure breed. Then Another case is if they are not pure breeds. So that is if they are TT, then we expect TT to be crossed with homozygous recessive like this. So the offspring are going to have TT. So in short, half of the offspring are going to be tall, then half of the offspring are going to be short. Then we'll tell that that person is a hybrid. So let me just bring you into context. So after we do the back crosses, and if we get all of the offsprings to be tall, then we can say that the father is supposed to be a tall person, a purely tall person. So if we find the father, we do, and we find this is a genotype, then we can be sure that it was the same father. But if some of the children are short, then we should expect the father to be a hybrid also. That's how to do back cross. Usually they come in paper 1. So there are some crosses and their expected outcomes. It's not important to master this, but if you do a lot of monohybrid crosses, you can know them. Okay, so these are all possible crosses and the possible outcomes. So if you want, you can master them. If you don't want, you can, of course, you'll be spending time to do the crosses to get the answers. Let's take a look at the last example. In maize plants, the allele M for white seeds is dominant over the allele M for yellow seeds. 100 heterozygous plants were self-pollinated and 250 yellow seeds were collected. What is the expected ratio of white seeds to yellow seeds? White seeds to yellow seeds. So the expected ratio, we have to do a crossing. So they have said 100 heterozygous, so we expect all these 100 to be to have capital M and small m. So they were self-pollinated, which means it was them with themselves. So MM, we're going to cross it with MM. Then we give them the expected ratio of white seeds and yellow seeds. Then what is the expected number of white seeds? We're going to use the same ratio and this number to determine the expected number of white seeds. What is the expected total number of seeds? We're going to add this 250 to what we're going to get in here. So let's get to it. So we're talking about heterozygous. So these are the parents. So when you do cross, this is what you're going to get. I'm not going to show you. We have consumed about 27 minutes. So MM, when you do the cross, if you're going to get these. So what is the expected ratio? So the expected ratio is usually on the phenotype. So this is white, this is also white, this is also white, this is yellow, because they are both recessive. So the expected ratio of white to yellow is 3 to 1. So question B, what is the expected number of white seeds? Expected number of white seeds. So we're going to use the ratio. Then question C was what is the expected total number of seeds. So white to yellow, in A we determined that it was 3 to 1. Sorry, having a dry throat. So the expected ratio is 3 to 1. But we have been told that we have 250 yellow seeds. So the number of white seeds we don't know, we're going to put x. So we do cross multiply, multiplying x and 1 and 3 times 250. So x times 1 it's x, and 3 times 250 it's 750. So we're going to have 750 white seeds. Question C, what is the expected total number of seeds? Expected total number, we add the 250 for the yellow and the 750 for the white. But let me just emphasize on this one, number of white seeds is 750. So now we know that number of white seeds is 750. Total is white seeds plus yellow seeds, like I said. So 750 plus 250, which gives us 1000 as the total number of seeds. So this marks the end of monohybrid crosses. I took time here because this is an interesting part of genetics, and I wanted you to have a feel of all the possible questions that can come. With that being said, Happy solving.